biogeography. So biogeography, this is the geographic patterns of species, distributions, either in a specific area or around the planet. So I'm going to show you one for a specific area. So this is like Darwin's finches. It's the same idea of Ecuador to the Galapagos Islands. Only in this instance, we're going from North America, one of these little mainland finches. You've probably seen something like this around in Ohio, to Hawaii. Now, this is a little bit farther trip. We're talking 2,000 miles away, but they're birds. It could happen. They could get blown onto, you know, a raft of trees or something like that, and then, you know, get blown out there and fly the rest of the way. Same sort of thing, though, happened when they got to the Hawaiian Islands. We get a huge variety of, instead of what we call finches, they call honey creepers. So honey creepers have fairly similar body types, but same sort of idea as far as the bills. The bills are very, very different, but some of the coloring is different as well. They're found at different habitats. So you can see wet forests versus specific slopes. Some of these are only found on one island on one side. As you can see, this is very specific. And their diets are very different from each other. And so this is an idea, this idea of biogeography, that these guys located and then spread out and distributed themselves into new species over time. And that can give us ideas on how that worked. So, you know, seeing that pattern of how they changed as they moved through the islands, how their habitat is reflected in the changes to the organism. Same thing with the finches. Another example of biogeography is the Australian marsupials versus the North American placental organisms. Well, North and South American, because we've got a South American one on here. But Australia is the only place on the planet that pretty much all of their mammals are marsupials, meaning that they have pouches for their young. We in the North American area, for North America, we only have one, which is the opossum. South America has a couple, but a majority of our mammals are what we call placental, meaning that they have their babies like we do, like humans. What we've got here are some examples of biogeography at play. So if we take a look, what do you notice about, say, for example, the sugar glider and the flying squirrel? Yeah, you probably noticed that they both have these long kind of flat tails. They both have the webs right here. They both have the little cute faces. And you may not have realized it, but they are on black backgrounds because they're both nocturnal. All right. These organisms look very similar to each other for a reason. Same thing for the anteater and the numbat. So they don't look identical, but they both have these long noses. If we could see it better at their feet, they both have digging feet. They have these long, bushy type tails. The Tasmanian wolf, which actually no longer exists, but it's the counterpart to the gray wolf here in North America. But they both look wolfy. I guess is the best description. It's just that the gray wolf is more furry, more poofy, but they have the same sort of general body layout. If we looked at their teeth would be very similar. And this is because even though they're found on different parts of the planet, right? So these guys are in North and South America. These guys are in Australia. They're also different groups of organisms. So they're mammals, but they're not closely related to each other. They still end up with a lot of characteristics in common because they have similar habitats. And so this is another way we can look at biogeography. Do you have, in similar habitats, do you have organisms that are showing up with similar characteristics to deal with those issues, right? So these guys are gliding around in trees to eat things like fruits and berries and nuts. These guys are both eating insects. These guys are both carnivores, right? So that's biogeography. Okay, our last line of evidence for evolution is experiments and field observations. So I'm going to show you a couple of these, and so that way you can see how they work as far as supporting the theory of evolution. So the first one is one that we've actually done by accident on golf courses. So I don't know how many of you are familiar with golf courses. I'm sure you've driven past one at some point. But on golf courses, there are three general different types of grass. So there is the rough, and this area is an area that gets mowed very infrequently. I know in the area that I live in, they basically don't mow the rough. Or if they do, they mow it like once a year. In this instance that they were doing the observations, they mowed the rough area once a month. So they went through and mowed down the grass in the rough once a month. The fairway is the central part of the golf course, and that is going to be mowed once a week. And then the putting green is right around the flag, and that gets mowed every other day at this specific golf course that they collected the data at. Now you can see up here, it's the same species of grass that was planted on the entire section, right? So whether it's the rough, the fairway, or the putting green, it was the same species of grass. And what they found after a couple of years is that even though it was the same species of grass, the grass had changed in the three different areas. All right. So on the area with the rough, the grass actually had very slow sexual maturation, meaning it took them a long time to produce seeds. And the area in the rough had the lowest seed production. So those plants made very few seeds. Compared to the putting green, and in the putting green, these plants grew very fast and matured very fast, meaning that they produced their seeds very early. So, they wouldn't have to grow very big before they started producing seeds. And not only that, each plant produced a lot of seeds. And then the fairway was in between. And so hopefully this makes sense. What's going on here to make the biggest difference between the rough and the putting green? So what happened to make the rough, or make the putting green have these fast maturing, high seed producing plants versus the rough with the slow maturing, low seed producing plants? And so what was happening is because these putting green plants were getting cut constantly, if you were a plant that didn't mature quickly, you didn't get to reproduce, right? And if you were going to reproduce, you better reproduce as much as you possibly can, because you might not get to reproduce again. Okay, so the plants weren't thinking this. This is just what ended up happening because of the high number of mowing that was happening on the putting green. All right, this is an experiment that has happened. Go back. By accident. So we developed antibiotics in the 1940s. That was the first time when penicillin was used to treat bacterial infections. So back in the 1940s, we would run a test to test the effectiveness of antibiotics. And so that's what you're seeing here in these plates. So here there's a little disc that has penicillin in it. And then you put bacteria on the plate and let them grow. And basically what happened in the 1940s, most of the bacteria would be killed. In this case, Staphylococcus. Same thing is true for things like strep. So these bacteria would be able to grow on the very edge, but this whole region in the middle would be an area of basically, no bacteria here. The bacteria would be killed. Now the kill zone in the central section of time, the kill zone is reduced. And today, we run the same experiment, next to no bacteria are going to be killed. And this is because we've ended up creating antibiotic-resistant bacteria. And this is because we have been using the antibiotics and not always in the ways that we should be. If you, for example, take your antibiotics and take them until you feel better and then stop, then you're going to end up with bacteria that are resistant, just like the pesticides that were talked about in an earlier lecture. And you're going to end up with a bacterial infection that is now partially resistant to that antibiotic. And so we've ended up creating these resistant bacteria that are resistant to not just penicillin, but we actually have staph bacteria that are resistant to more than one antibiotic. If you've heard of Mrsa, it doesn't stand for multiple, it stands for methanicillin, which is another type of antibiotic. But there are strains of Staphylococcus that are multiple resistant to antibiotics because of this process. And then the last one is our fruit fly experiment, right? We can create these kinds of experiments to be able to see these changes in populations over time. All right, and then I have this last question for you. Asked this question at the beginning, so I want to ask it here again at the end. Right. And that is the end of Chapter 10 and Natural Selection.